this would be the question 25, part 2 video. Okay, so for part 2 video, we will solve the questions B and C. Yeah, B and C. So for finding the solution for question A, yep, so you can refer to the question 25, part 1. Yeah, part 1 solution video. Okay, so just now in the part A, right, in the part 1 video, right, so we have done with this, okay, by looking into the recombinant frequency. Okay, so now we look at question B. Explain how to determine whether S gene and M gene are linked. Yeah, S gene and M gene are linked. Yeah, okay, so you can see here, this is how we test whether the P gene and also the S gene are linked or not. And this part, right, for this part is basically we are testing whether the P gene and the M gene are linked or not. Okay, so now the question is asking you, how are you going to determine whether the S gene and the M gene, whether the S gene and M gene are linked or not? Okay, so what we should do, actually, you can just copy back how it is done. Okay, in order to test whether the two genes are linked or not, so yeah, you need a heterozygous. You need a heterozygous, yeah, heterozygous dominant. Heterozygous dominant. Okay, so for example, you see purple and smooth. Purple and smooth are both dominant. Purple and smooth, yeah, both are dominant. So you need a heterozygous dominant, okay, in order to test. Okay, so in this case, you want to test S gene and M gene. So that means that you have to find a plant that is heterozygous dominant for S and heterozygous dominant for M. That's the first parent. Then after that, you will do the test cross. You see that? It will test cross, yeah? Homozygous recessive individual. It will test cross with the yellow. Yellow is actually recessive. Okay, and then shrunken seed is also recessive. So in other words, you are doing the test cross. Okay, so that's why, yep, in order to determine whether the S gene and the M gene are linked or not, so this is what we should do. Okay, so again, same thing, you have to determine the parent first. Yeah, you have to determine the parent first. Yeah, okay, so that's why for B, yeah, so for the parent, yeah, for the parent, okay, you want to determine for S gene and M gene. So that will mean that for parent must be heterozygous. One big S, one big M, and then one small s, one small m. This is the parent requirement. Okay, then you will do the test cross. So you will do the test cross. So small s, I should write in linkage way, yeah. So SM and SM. So this is the condition. Yeah, this is the condition. Okay, so of course you might ask, can I find a parent that is big S, small m, then slash small s, big M? Can or not? Yes, can, because it's still heterozygous, right? It's still heterozygous. Okay, so for this, I will just state this lah. Yeah, so this is the parent situation. Okay, so since the question is asked you to explain how, yeah, explain how you want to do it. Okay, so you cannot list it like this, so you have to state. Okay, so you will do the heterozygous, okay, plant, right, that is having big S is smooth. Yeah, big S is smooth. Okay, and then big M would be starchy. Okay, seed plant, yeah. So you will take a plant that is heterozygous, smooth and starchy seed, yeah, and then you will do the test cross. Yeah, you will do the test cross. Okay, test cross with which individual? With this individual. This individual that is homozygous. Okay, it is homozygous. Okay, homozygous. Small s would be shrunken. Okay, and sweet, yeah. Seed plant. So this is what you should do. You should do the cross first. You should do the test cross first. Okay, so after you do this test cross, then again, we do the same thing. We will check the recombinant frequency. If the recombinant frequency is less than 50%, then that means that this S gene and the M gene are linked. But if, let's say, the frequency is equals to almost 50, or it is larger than 50, which is normally not possible, okay, then yep, it will mean that it is actually considered as the non-genetically linked. Okay, so that is basically how we solve B. Yeah, that's how we solve B. Okay. So now let's say you have done this test, yeah? Okay, so question C state that if you have done this test and you find out that the S gene and M gene, they are linked and their distance is 41 centimorgan. Centimorgan means map unit and map unit means recombinant frequency, yeah? Okay. So S gene and M gene, yeah, you have do this cross already, okay, and you find out that the S gene and M gene are linked together at 41 centimorgan. That means that they are genetically linked, right? Okay, 41 means they are genetically linked. Okay, so they are asking what is the distance between P and M gene? Yeah, P gene and the M gene. Okay, so now we have to list down all the info that we have, yeah, for this type of question. Yeah, question C. Okay, so from what we know just now, okay, from here, the question told us that S gene and M gene is 41 centimorgan. Okay, so that means that the distance between S to M, yeah, the distance between S to M is 41. Yeah, it's 41 centimorgan. So centimorgan, I write like this, yeah? Not the cm, this is centimorgan. Okay. So next, what else we know? From the question just now, we have calculated the distance between the P gene and S gene. Yeah, the P gene and S gene, right? P gene and S gene, their recombinant frequency is 21.8%. Yeah, the recombinant frequency is 21.8. So that means that on the chromosome, the distance between the P gene and the S gene is 21.8. Yeah, it's 21.8. Okay, so that's why, yeah, this one we can use here also. Okay, so the distance between the P gene and the S gene, yeah, on the same chromosome is 21.8 centimorgan. That's a distance. Okay, then the distance that we calculate just now, the one that is between P and M, yeah, the one that's between P and M is 49.6. So it's like almost 50 already. It's almost 50. Okay, so P and M gene, that means that they are on the same chromosome, but they are too far apart. So when they are too far apart, the recombinant frequency, right, the ratio when you do the test cross, it will be 1 to 1 to 1 to 1. It will be similar to the Mendel's model, the normal Mendel's model, 1 to 1 to 1 to 1. Okay, so it's give you exactly almost 50%. So meaning that means that their distance on the chromosome is exactly 50? No. Okay, no. It only means that the distance between them on the chromosome is more than 50. It's larger than 50 for sure. Okay, so that's why they will give you this type of a recombinant frequency. Okay, so because of this, what we know is just that between the P gene and the M gene, okay, what we know is that they are having more than 50 centimorgan. Yeah. If you still remember in the mind map, I got mentioned, yeah, if let's say the distance is larger than 50 map unit, then the recombinant frequency will always show as 50. Yeah, if you still remember, hope you still remember that in the tutorial as well. Okay, so if let's say the distance is larger, yeah, the map distance is larger than 50, then the recombinant frequency will always shows maximum at 50 only. Okay, so that's the example here. Yeah, you can see here. All right, so we come back here. So we know it's more than 50. Okay, so that's why now they want us to find the distance between the P gene and the M gene. They want us to find the distance between the P gene and M gene. They want to find the exact one. Yeah, the exact one. Okay, so you have all the number here. Then of course you have to find the possible sequence. Yeah, you have to find the sequence. Yeah. Okay. So first thing first is we take the largest number would be this one because this one is uncertain. So we will take the largest number, which is S and M. Yeah, S and M. Yeah. So S and M is 41. Yeah, S and M is 41. So we will write that. So the distance between S and M. Yeah, the distance between S and M is 41. Lengthen it a bit. Okay, the distance is 41 Mach unit or 41 centimorgan. Okay, so this one, yes. So second, the distance between P and S is 21.8. Yeah, 21.8. So P and S, yeah. So that means that the P and S, it can be P can be here. P can be here also. Okay, it's either one. 28.1. We don't know. We don't know whether this P gene is to the right side of the S gene or this P gene is to the left side of the S gene. We are not sure. Okay. Then lastly, if let's say this P gene is to the right side, then that means that between the P and M, yeah, between the P and M is actually around, you take 41 minus 21, yeah? So it's around like 19, 19.2, around that. Okay, if let's say the P gene is towards the right side of the S, yeah, then the distance between P and M should be around 19.2. But obviously, it's not. Why? Because we already know P gene and M gene is more than 50. It won't be 19.2. Okay, if it is 19.2, then it will be shown in the recombinant frequency already. Okay, so that's why it is impossible that the P gene and M gene is 19.2. Yeah, it's impossible. Okay, it's impossible. So because of this, then yeah, it's 19.2. But obviously, based on the calculation just now, we know it's more than 50. Yeah, so that's why this is definitely out. Okay, so if this is definitely out, so that means that this is no. Okay, so that means that the P gene must be towards the left side of the S gene. P gene must be towards the left side of the S gene. Yeah, so now we can confirm. Okay, no need to put question mark already. So we now can confirm here. So after we have confirmed, okay, from P gene to S gene is 21.8. From S gene to M gene is 41. Can we find the distance of the P to M? Yes, we can. Okay, so the distance P to M, yeah, the distance from P to M gene, from P gene to M gene, it will be equals to, yep, you sum it up. P gene to M gene, it will be 21.8 plus 41. So it's 21.8 centimorgan plus 41 centimorgan. Okay, so with that, the distance is 62.8 centimorgan. Okay, so that means that the distance between P gene and M gene is actually 62.8 centimorgan. It is larger than 50. So that is why when giving the recombinant frequency, they are giving 50%. They are showing the ratio of 1 to 1 to 1 to 1 in their offspring. That is the reason, because they are too far apart. They are more than 50. So that's why. Okay, so that's why with that, we can now confirm this is the correct answer. The distance between the P and M is 62.8.